So let's take a look at your lipid metabolism. Okay, and so you have here your lipid metabolism. Okay, and so let's take a look at this one. Okay, and so of course, and so all lipids are derived from your acetyl coenzyme A. No, and so your all your lipids are derived or what you call this or in whole from your acetyl coenzyme A. No, and so your acetyl coenzyme A here, and so this one is the substrate in the synthesis of fatty acids, and so terpenes and your steroids. Your steroids here, and so uh, representative of that is your cholesterol. For your terpenes, you have therefore your beta carotene and the rest, and then of course this one, of course. When your cells require energy, and so your fatty acids are degraded, and so remember that your acetyl CoA, and so this one is used in the production of energy in your citric acid cycle. Okay, and so when cells require energy, and so your fatty acids are degraded, and so to produce acetyl coenzyme A. Of course, this one, and so is diverted into the citric acid cycle. Okay, and so that is for the connection. Okay, moving on. And so your exogenous pathway. And so let's take a look at this one. Your absorption of your triglycerides and your lipid nutrients and their distribution in the body. And so that is via your uh, lipoproteins. That is for your exogenous pathway. Okay, and so your absorption of triglycerides and distribution via your lipoproteins is your exogenous pathway, while for your endogenous pathway, and so that is the transport of your lipids produced by the liver, of course. And so in your exogenous pathway, you have here your what you call this dietary source, dietary triglyceride. Okay, and so you have there the absorption. And then your endogenous pathway, and so this one is a what you call this, it involves the lipids that are synthesized by the liver. Okay, and so you have there your fatty acids, and so they are stored in your triglyceride, and most, and so you have here your triglyceride here, they are the most concentrated energy source accordingly. And so as what we talked last time, that your triglycerides, and so they are more efficient in storing energy comparing it to your glycogen. Okay, and so with this, let's take a look at your insulin. And so the function of your insulin in your triglyceride, in your lipid metabolism. Your insulin, and so it promotes triglyceride storage by inactivating sensitive lipase. Ayan, so by inactivating your sensitive lipase here. And so that is with that. Okay, and so it inactivates your hormone sensitive, and so this lipase here, and so it is sensitive to your insulin. Okay, and so you have there with that also, it activates your triglyceride synthesis in fat and muscle cells for your insulin. Okay, and so also your insulin stimulates your release of VLDL also. Then, and so in case that you have your blood sugar levels or your blood glucose levels fall, and so insulin levels is actually decreased, no? And so because, and so you need more of your energy, that is why you're not going to store, you're going to break down to produce energy for this. And so in case there is a decrease in your blood glucose levels, and so that is why instead of your insulin is increasing, you have the enzyme that is you have your hormone, sorry, your hormone glucagon, that is increased in level in the concentration. Okay, and so with this, and so your glucagon will stimulate your lipolysis accordingly. Okay, and so that is for that. Okay. Okay, and so your triacylglycerol. And so this one, your triacylglycerol synthesis, and so it regulates the level of fatty acids that are available for the body for energy generation, no? And so in synthesis of molecules like your phospholipids. Okay, and so your triacylglycerol or your triglycerides are hydrolyzed in the adipocytes. Okay, and so of course your triglycerides are constantly, and so both they are synthesized and hydrolyzed, no, and so to fatty acid and glycerol. Okay, and so you have there, and so of course with this, and so your what you call your hydrolysis, and so will release small fractions of fatty acids in the blood. And then of course these fatty acids and so are what you call this transported into the liver, and so wherein they are incorporated to your triglycerides. Okay, and so and then of course they are packed to VLDL. Okay, and so you have there they are packed to VLDL. Okay, and so of course, is cycling system here, and so the cycling system, and so it ensures the sufficient that what we call the sufficient fatty acids are available for the body's energy and biosynthetic requirements. Okay, and so your excess fats, and so if there is are actually excess fats, and so they are re-esterified into triglycerides. Okay, and so because relatively, and so when you have excess fatty acids present, and so you have here they have toxic effects on cells, no? And so that is why you need to resterify it, and so into triglyceride. Okay, and so moving on. Okay, and so this one is an illustration of your what you call this synthesis of your fatty acids. Okay, and so synthesis of fatty acid, synthesis of triglyceride, and then release of fatty acid. Sorry, and so you have there. So let's take a look at your glyceroneogenesis. And so this one is the synthesis of your glycerol. Okay, and so your glycerol 3-phosphate is synthesized from glucose or glycerol or other substrate. Okay, and so your glycerol 3-phosphate is required to synthesize your triglyceride. And so note that you have it as your triacyl glycerol, and glycerol is part of your triglyceride. Okay, and so your pyruvate decarboxylase and your phosphoenol pyruvate kinase are the important enzymes that are needed in your glyceroneogenesis. Okay, and so again, your glycerol, this one is part of your triglyceride. Okay, okay, and so let's take a look at your lipogenesis. Okay, and so in this one, your lipogenesis or your triglyceride synthesis, and so this one involves a three-stage of acetylation. And so you have first one, of course, you have your glycerol 3-phosphate, and so reacts with your three molecules of your acyl CoA, of your acyl coenzyme A. And so with that, and so to produce your lysophosphatidic acid, and this one is catalyzed by glycerol 3-phosphate acyl transferase. And so the other one, your acyl dihydroxyacetone phosphate. And so with this, and so produces your lysophosphatidic acid. Okay, and so your acyl dihydroxyacetone phosphate produces lysophosphatidic acid, and this one is catalyzed by acyl dihydroxyacetone phosphate reductase. Okay, and so you have here and then your lysophosphatidic acid, and so is converted to phosphatidic acid that is catalyzed by acyl transferase, and so that is catalyzed by acyl transferase. Okay, and so with that, and then of course you have here your lysophosphatidic acid, and so it utilizes a an NADH or an NADPH. Okay, and so you have there. And so let's take a look at this one. And so your phosphatidic acid, and so is utilized in the synthesis of phospholipids. Okay, and so you have there your phosphatidic acid. This one is converted to diacylglycerol, and this one is catalyzed by phosphatidic acid phosphatase, and your phosphatidic acid here, and so is utilized in the synthesis of phospholipids. Sorry, and so you have there, and then and so once formed, and so you have your phosphatidic acid, and so is converted to diacylglycerol by your phosphatidic acid. Okay, and so by your phosphatidic acid phosphatase. Okay, and so your diacylglycerol, your phosphatidic acid is converted to your diacylglycerol, and then your diacylglycerol is converted to your triacylglycerol, and so this one is catalyzed by your diacylglycerol acyl transferase, no? And so this one is what they call this your diacylglycerol, sorry, and so can be converted. into monoacyl glycerol. Okay, and so this one, your diacyl glycerol can be converted into monoacyl glycerol. Of course, that is by your acyl transferase. Okay, and so giving you, of course, your triacyl glycerol already with you. So moving on, let's take a look at your hydrolysis of your triacyl glycerol. And so, or this one is otherwise known as your lipolysis, of course. And so, when your energy reserves are low, and so your body's fat stores, and so that is your triacyl glycerol, and so they are mobilized in a process, and so that is your hydrolysis or your lipolysis, no? And so you have there, of course, your lipolysis. And so lipolysis in the adipose tissue, and so happens during periods of fasting and vigorous exercise, and so, and of course in response to stress. Okay, and so you have there. Accordingly, you have hormones, and so bind to your specific adipocytes, and so to your specific adipocyte plasma membrane receptors. Of course, in your adipocytes, you have there your your membrane receptors for your hormones. And so this one, and so when they bind, and so it will start, it will begin the reaction sequence. And so again, and so your in your adipocyte, and so you have there your receptors for your hormones that are needed to break down, and so that will stimulate the breakdown of your triacylglycerol. And so examples of hormones, you have your catecholamines, your epinephrine, and your norepinephrine. And so these hormones will bind to this specific receptor present in your adipocyte membrane, and then with that, and so it will start your a reaction, and so a series of reaction. Okay, and so you have there. Of course, and so the effects of this binding of your hormones to the receptor in your adipocytes, and so you have there, it will increase your cytoplasmic cyclic adenosine monophosphate levels. Okay, and so this cyclic adenosine monophosphate levels will activate the phosphorylation of a protein called as perilipin A. And so again, and so your hormone, your what you call this, a binding of your hormone to the receptors in the adipocytes will increase your cytoplasmic levels of your cyclic adenophosphate, uh, ad cyclic adenosine monophosphate. And then this cyclic adenosine monophosphate will activate your phosphorylation of your perilipin A. And so, in what is this perilipin A? And so that's one, and so it prevents your lipase access to your triglyceride. Okay, and so when your perilipin A is phosphorylated, and so there is a change in the conformation of this protein, and so thereby, and so what happens here is uh it will expose, and so the phosphorylation of this one will cause the a conformational change, and this conformational change will cause your perilipin A to expose your triglycerides to this to your lipases. Okay, and so you have there. Okay, and so your perilipin A, and so regulates your lipid storage by coating your adipocyte lipid droplet. Of course, and so with that, it coats, and so it prevents your lipase access. And so that is why when what you call this, when there is a conformational change of your uh perilipin A, and so it will expose your triglycerides to your lipase, and this lipase, of course, will do its job. Okay, and so you have there, and so actually that is the first step in your hydrolysis of your uh in the hydrolysis of your triglycerides. Of course, that is you have there your catalyzed by your adipose triglyceride lipase. Okay, and so you have there. Okay, and so your lipolysis. Okay, and so your perylipin A already is phosphorylated, and then of course it already exposed your triglycerides, and so giving an access, and so for your adipose triglyceride lipase to act on. Okay, and so with this, your adipose triglyceride lipase will convert your, will hydrolyze your triglyceride to produce your diacylglycerol and fatty acids. Okay? And then your diacylglycerol, ayan, so is further hydrolyzed to produce monoacylglycerol and fatty acid. And so this one is catalyzed by your hormone sensitive lipase. And then of course, for the final reaction of your hydrolysis of your triglycerides or in your lipolysis, and so you have there the conversion of your monoacylglycerol to your glycerol and fatty acid. Okay, and of course, this reaction is catalyzed by monoacylglycerol lipase. Okay, and so but wait, and so you have here your insulin again, and so your insulin, and so it will suppress the synthesis of adipose triglyceride lipase. Okay, and so it will suppress the synthesis of your adipose triglyceride lipase and will also decrease the intracellular activity of your cyclic adenosine monophosphate. And so that is actually a control of your lipolysis still. Okay, and so of course, and so after you have already produced your glycerol and fatty acids, and so this glycerol and your fatty acids are released into the blood. Okay, and so they are released into the blood, and so of course, and to the circulation. And so your glycerol, and so that can be used as energy. And then for your fatty acids, and so it will go into your, it will be, yes, and so utilized, and so still for the production of your energy. And so let's take a look at this later as we go on. Okay, and so you have your degradation of your fatty acid. Okay, and so accordingly, the fatty acids are degraded by sequential removal of two carbon fragments. And so from the carboxyl end of your fatty acids, no, and so thus in your degradation of your fatty acids, with this it involves the shortening of your chain by one carbon through oxidative decarboxylation. And so under your degradation, you have actually your uh, beta oxidation, and then you have your omega. Your omega oxidation here is a minor pathway, and this one, and so it does not what they call this minor pathway in humans, and it rarely occur in humans accordingly. And so but then let's take a look, and so your omega oxidation, the terminal methyl group in your omega oxidation accordingly, and so the terminal methyl group with this one is subsequently oxidized to your carboxylate group. And then of course after that, after it is oxidized, and so after it is oxidized, and so it will be shortened, and so the fatty acid is shortened through beta oxidation. And again, and so sabi natin kanina, this one is a minor pathway. And so that is a minor pathway in humans, and it rarely occur in humans. Again, and so let's take a look in your omega oxidation, and so the terminal methyl group is converted into an alcohol, no, and so this one is converted into an alcohol, and this alcohol is subsequently oxidized to produce your carboxylate group, no, and so it is subsequently oxidized to produce your carboxylate group. And then of course, and so that is catalyzed by your the sequential reactions with that is catalyzed by your aldehyde dehydrogenase. Okay, and so and then of course the it will further be shortened via your beta oxidation. Okay, and so but then with that, and so let's take a look at your beta oxidation. And so in your beta oxidation, you have actually this four steps. And so you have there your dehydrogenation with your FAD. Ayan, your flavine adenine dinucleotide. And then you have your reversible hydration. And then your dehydrogenation with NAD and reaction with coenzyme A. And so with this, and so your reaction with coenzyme A produces acetyl-CoA and a fatty acid derivative of your coenzyme A. And so, which of course, they are shorter by 2 carbon atoms. Okay, and so let's take a look at this one. Okay, and so your entry to your entry of your fatty acids into your mitochondria, of course, first, and so it should be activated. And so your fatty acid should be activated first into acyl-CoA. And so by your acyl-CoA synthetase. And then this acyl coenzyme A is converted into your acyl-carnitine derivative. And so note, when you already have your acyl-CoA, and so it cannot pass through your inner mitochondrial membrane. And so in the outer mitochondrial membrane, and so that is where your activation occurs. And so your activation of your fatty acids into your acyl-CoA. And so that is catalyzed by acyl-CoA synthase. And so once there is an acyl-CoA, and so note that your inner mitochondrial membrane is impermeable to acyl groups. And so with that, you need a carrier. Okay, and so you have here your carrier protein, and so you have here your acyl-CoA is converted into acyl-carnitine derivative that is catalyzed by carnitine acyltransferase 1. Okay, and so you have there. And so your carnitine here, and so this one is used to transport acyl group into the mitochondria, into the inner mitochondrial membrane. Okay, and so into the mitochondria. Okay, and so so again, your carrier protein will transfer the acyl-carnitine in the mitochondrial matrix, and then your acyl-CoA is regenerated by carnitine acyltransferase 2, and so your acyl-CoA is released, and then your carnitine is transported back into the membrane, intermembrane space. Okay, and so let's take a look at that in this illustration. And so again, your fatty acids are activated to form your acyl coenzyme A. And so that is catalyzed by your acyl CoA synthetase. Ayan, so that is found in the outer mitochondrial membrane, no? And so your acyl CoA, and so again, and so the inner membrane, and so you need this acyl CoA, you need it to enter the mitochondria. But then the inner membrane is impermeable to your acyl groups, and so that is why you need a carrier accordingly. And so you have your carnitine, and so your acyl CoA, and so will react with your carnitine again, and so to form your acyl carnitine derivative. Okay, and so you have there your acyl carnitine derivative. Okay, and so with that, and so your what you call this, the reaction is catalyzed by your carnitine acyltransferase 1. Okay, and so your acylcarnitine is transported across already of your inner membrane, and so your the carnitine is already inside your mitochondria. Okay, and so again, your acylcarnitine is transported across the inner membrane by your acylcarnitine translocase. And then, and so this acylcarnitine is reconverted to your carnitine and acyl coenzyme A. A. Okay, and so by your carnitine acyl transferase 2. Okay, and so with that. Okay, and so your carnitine, and so will move, and so it will go back into the into the outer membrane. And so note that your acyl carnitine translocase is an anti-porto protein. And so with that, and so it transports one acyl carnitine into the mitochondrial matrix for every carnitine that is transported into the intermembrane space accordingly. And so with that, of course, your fatty acid in the form of your fatty acid is already inside your mitochondria in the form of your acyl coenzyme A. Okay, and so then of course you can already do your what you call this, your degradation of your fatty acids again. And so you have there your beta oxidation. Okay, and so as what I said earlier, your fatty acid is activated by reacting with ATP and then coenzyme A, and so to produce your acyl CoA. And so, and that is catalyzed by your acyl CoA synthetase. Okay, and so, and that is transferred to the inner mitochondria by your carnitine, and then your acyl coenzyme A. Your carnitine, this one is the carrier protein accordingly. Okay, and so your acyl CoA, and so once inside the mitochondria, your acyl CoA is converted into your trans alpha beta enol coenzyme A. Note that this one is for your saturated fatty acids. Okay, and so your acyl CoA, and so is converted to your trans alpha beta enol coenzyme A. And so this one, this uh, reaction is catalyzed by acyl CoA dehydrogenase. Okay, and so with this, accordingly, there is one hydrogen atom is removed from your alpha and beta carbons. And so what is your alpha carbon? And so that is the carbon that is next to your carbonyl carbon. Okay, and so and then the beta that is next to your alpha carbon. Okay, and so with this in your acyl CoA to your trans alpha beta enol coenzyme A, and so there is one hydrogen atom is removed from both your alpha and beta carbons. And so, and this hydrogen, and so saan sila mapupunta? And so they are transferred to your FAD, your fladenine adenine dinucleotide. Yes, and so forming your FADH2. And so note that you here in your beta oxidation, you are trying to produce energy. And so saan mapupunta itong FADH2? And so it will go into your electron transport chain. Okay, and so it will go into your electron transport chain. And so let's take a look at the next reaction. And so for the second reaction, and so this one, and so your trans alpha beta enol coenzyme A, and so is converted to beta hydroxy acyl coenzyme A. Okay, and so again, your trans alpha beta enol coenzyme A is converted to your beta hydroxy acyl coenzyme A. And this one, this reaction is catalyzed by your enol coenzyme hydrase. This one is your what you call this your uh the conversion of your trans alpha beta enol coenzyme A to your beta hydroxyacyl coenzyme A is catalyzed by your enol coenzyme A hydrase. And so with this, and so it involves a hydration of your double bond. And so note that in the removal of your hydrogen in the first reaction here, and so in the removal of your hydrogen, and so in your alpha and beta carbon atoms, and so there is already a formation of a double bond. Okay, and so because and so it is not you removed one hydrogen in each, then thereby giving you a space for an addition reaction, forming a double bond. Okay, and so double bond between your uh, alpha carbon and your beta carbon. And so there is already a double bond. Okay, and so in your second reaction here, and so there is a hydration of the double bond. And so there is a hydration of your double bond that is formed in your alpha and beta carbon. Okay, and so with that, and so your beta hydroxyacyl coenzyme A, and so is converted into your keto acyl coenzyme A. And so your beta hydroxyacyl coenzyme A is converted to your beta keto acyl coenzyme A that is catalyzed by your beta hydroxyacyl coenzyme A dehydrogenase. Okay, and so that is catalyzed by your beta hydroxy acyl coenzyme A dehydrogenase. And this one, there is a formation of your NADH2 here. Okay, and so and then you have your and this one, and so of course you have what you call this you there is a there is a hydration. Okay, and so in the next in this one, in this reaction already, and so your hydroxyl group is oxidized. Okay, and so your hydroxyl group in your beta hydroxyacyl coenzyme A, and so your hydroxyl group is oxidized, and so to form your beta ketoacyl coenzyme A. Okay, and so that is already oxidized. The hydroxyl group is oxidized. Okay, and so giving you, and so with this, there is the formation of your NAD, formation of your NADH2. Okay, and then you have your beta ketoacyl coenzyme A here uh will react with another coenzyme A, and of course will combine with your another coenzyme A, and the formed one is cleaved, and so to the condensed one is then cleaved to acyl coenzyme A and your acetyl coenzyme A by your beta ketoacyl coenzyme A thiolase. Okay, and so first in this fourth reaction here, and so your beta ketoacyl coenzyme A will combine with another coenzyme A. And then after that is combined, and so the resulting product is cleaved. And so it will be separated, and so to produce your acyl coenzyme A and your acetyl coenzyme A. This reaction is catalyzed by your beta ketoacyl coenzyme A thiolase. Okay, and so that is for the process. Previously, and so we already saw your four steps in your beta oxidation, and so we already saw your four steps in your beta oxidation. And so at the last step, and so we cleaved, and so we produced one acetyl coenzyme A and an acyl coenzyme A. Note that your acyl coenzyme A, and so this one is still, and so your acyl coenzyme A is still made up of several carbon atoms. No, and so your acetyl coenzyme A, that is cleaved, and so that is made up of two carbon atoms. Okay, and so in the acyl coenzyme A will undergo again the sequence of the series of your beta oxidation steps, and so it will go again, and then until it will release another acetyl coenzyme A. Okay, and then the cycle will be repeated until it will release again another coenzyme A, until, and so the acyl coenzyme A, and so has four carbon atoms already, until it produce, and that four carbon atoms, and so that will be cleaved, and so to produce two acetyl coenzyme A. And so in case, let's take a look in case of your palmitic acid, and so activated to palmityl coenzyme A, and so that is a 16 carbon atoms. And so isipin mo, and so first cycle, you remove acetyl coenzyme A, two carbon atoms na yon. And so how many carbon atoms are left in your palmityl coenzyme A? You already have 16 minus 2, then you have 14. The acyl co A is 14 carbon atoms. And so what will you do with that? And so it will go again. And so the beta oxidation process, the cycle. And so it will go again. And so until it will until it will be reduced into two carbon atom less. And so it will be reduced. And so one acetyl coenzyme A will be removed again. And so in that, and so 14 minus 2, then you have 10 carbon atoms more, and so it will undergo again your steps in beta oxidation, and so that is actually known as your beta oxidation spiral, no? And so it will undergo again the steps of your beta oxidation, and so you have here until it will become 8, until it will become 6, until it will become 4. On that fourth, and so your 4 carbon atoms will be cleaved into 2 acetyl coenzyme A. Okay, and so you have 2 carbon atom less. Okay, with that. Okay, hanggang actually, and so until you will produce 2 acetyl coenzyme A. Okay, and so that is for your what they call this That is what we call that is your beta oxidation. Okay, and so it will be repeated, and so the steps in your beta oxidation is repeated until, and so you have a 4 carbon acyl CoA that is then cleaved to produce 2 molecules of acetyl coenzyme A. Okay, and so with that in mind, and so please note that the produced NADH and FADH as per cycle of the beta oxidation. Okay, and so if you have longer, and so know that if you have uh longer fatty acid chain, then you will produce more of uh you will yield more of energy. Okay, and so with that, and so but that for example of this, your palmitic acid, and so your palmitic acid, and so let's take a look at your energy yield in the follow. I mean, in a bit. Okay, and so with that. Okay, and so this one is an illustration of your 4 steps. Okay, and so of your beta oxidation. Okay, and so your acyl CoA and then your acetyl coenzyme A as the product. Okay, and so note that your acetyl coenzyme A and so is produced, and so thereby it will enter your what you call this, your citric acid cycle again for the production of energy. And so your FADH, and so the fate of this ones, your FADH and your NADH, and so they will enter your electron transport chain. Okay, and so in your beta oxidation, and so the rate of your beta oxidation accordingly depends on the availability of your substrate. And so in the muscle, and so it is dependent on the availability of your fatty acids, and of course you have therefore your the energy requirement of the tissue. Okay, and so if you need more, and so if the tissue need more energy, and so and glucose is depleted, then of course, your fatty acid will be degraded. Okay, and so your high NADH and or your NAD, and so ratio, and so will what you call this inhibit your beta hydroxyacyl coenzyme A. Okay, and so it will inhibit your beta hydroxyacyl coenzyme A dehydrogenase. Okay, and so it will inhibit your beta hydroxyacyl coenzyme A dehydrogenase. Okay, and so and then your high acetyl coenzyme A here, and so high acetyl coenzyme A levels will decrease the activity of your thiolase, and so the activity of your thiolase. Okay, and so your thiolase here, and so this one is the enzyme that will break down, and so that is involved actually in your fourth reaction of your beta oxidation. Okay, and so you have there, and so in the liver, and so your beta oxidation naman depends on the transport of your phospholipids and triacylglycerols into the mitochondria. Okay, and so sorry, you also the synthesis. Okay, and so you have here your your beta oxidation, and so depends on the uh, transport of your phospholipids and your triacylglycerols to the mitochondria. And then of course, and so in the liver, and so there is the what you call this conversion of your excess glucose into your fatty acids. Okay, and so you have there, and so with this, and so your when of course there is a glucose when there is an excess glucose molecules, and so they are converted into your fatty acids, so and so of course your malonyl coenzyme A there, that is the uh first product, and so that is committed for the synthesis of your fatty acids. Okay, and so let's take a look at the others for okay, and so let's take a look at your what you call this energy yield of your fatty acid oxidation, no? And so your oxidation of your fatty acid generates large number of ATP molecules. Okay, and so for this, and so your FADH and your NADH that are produced, and so in the reaction, and so will enter your electron transport chain and your oxidative phosphorylation. And so with this, and so you will still you can still utilize, and so the conversion that we used in your that we utilized in your uh glycolysis and your citric acid cycle, no? And so that we utilize in there. And so, but then for this, and for my reference for this, and so you have your 1.5. Accordingly, and so 1 FADH is equivalent to 1.5, and your 1 in ADH is equal to your is equal to 2.5 molecules of ATP. Okay, and so let's take a look at the yield of this oxidation of your palmitic acid, and so of your palmitoyl CoA. And so note that your palmitic acid, this one is the most common saturated fatty acid in humans. Again, and so this one is your palmitic acid is the most common fatty acid present in humans. Most common saturated fatty acid in humans, of course. And so this one, and so your palmitic acid, and so is converted into your palmitoyl coenzyme A. And the formation of palmitoyl coenzyme A, and so uses 2 ATP. And so of course, you know the one to subtract. And so the 2 ATP that is used in the formation of your palmitoyl coenzyme A, and so from your palmitic acid. Okay, and so with that, and so in the reaction, and so you produce 10 you produced 7 FADHs, no? And so you produce FADH, and so you produce 7 FADH, and then also you produce 7 NADH, and so you also produce 7 NADH, and then you produced 8 acetyl coenzyme A accordingly. And so from 1 mole of your palmitoyl coenzyme A, and so that is produced from your palmitic acid, and so you produce 8 acetyl coenzyme A. And accordingly, and so 1 acetyl coenzyme A will produce 10 ATP. And so if you try to take a look at this one, comparing, if you try to sum it all up, and so that produce ATP here, and so it is 1 mole, and so is 1 mole of ATP, and so will produce more, 1 mole of your uh, fatty acid will produce more energy comparing it to your 1 mole of glucose accordingly. And so that is for the energy yield. Okay, and so that is for the energy yield of this. Okay, and so and so please take a look, and so at the comparison between the energy yield of your glucose and your fatty acid here. This. Okay, and so in your ketogenesis, and so of course you already produce your uh, acetyl coenzyme A with you. And so you have your acetyl coenzyme A with you, and so with that it will enter your Krebs cycle. Okay, and so your ketone bodies here, and so your ketogenesis, and so your excess acetyl coenzyme A is converted into your acetoacetate, your beta hydroxybutyrate, and your acetone. And so these three here are actually your uh ketone bodies. And so in your ketogenesis, and so that is the formation of your ketone bodies. And so you have there. And so note that most of your acetyl coenzyme A produced during your beta oxidation, and so that is used by your citric acid cycle. And so other than your citric acid cycle, you have here the acetyl coenzyme A is utilized also for in the isoprenoid synthesis. Okay, and so you have there, and so under normal conditions, and so your fatty acid metabolism is regulated, no? And so that is regulated so that, and so small amounts of excess acetyl coenzyme A is produced. And so bakit? And so why is it that it should be small amount only? Because and so if you have much of excess acetyl coenzyme A, and so it will what you call this, it will undergo, I mean it will produce ketone bodies. Okay, and so you have there. And so in normal conditions, and so your fatty acid metabolism is regulated, that only small amount of excess acetyl coenzyme A are produced, and so thereby preventing accumulation of the intermediates of this one. Okay, and so you have there. Okay, and so you have there, and so if you have excess, much of excess acetyl coenzyme A, and so your ketogenesis will take place. Okay, and so let's take a look at your ketonemia. And so when you have actually a ketonemia, you have here an elevated amount of ketone bodies in the blood. Ayan, so in your ketonuria, ayan, so you have the presence of ketones in the urine. Of course, this one, your ketonuria will take place when you have here, when the ketones, ayan, so are excreted in the urine. And so, of course, it will always reflect, no? Ayan, so what is present in your circulation will reflect almost always in your urine. Okay, and so let's take a look at this one. Your ketogenesis, and so your ketogenesis occurs in the matrix of your liver mitochondria. Okay, and so of course this one, and so it begins with the condensation of your acetyl coenzyme A's, and so you have the excess, no, and so you have more of excess, you have an increased elevated acetyl coenzyme A, and so with that, and so you have a condensation of many acetyl coenzyme A to form your acetoacetyl CoA, and so you have your condensation of your you have your 2-acetyl coenzyme A, and so to form your acetoacetyl CoA. And so this one is catalyze your condensation of your 2-acetyl CoA to form your acetoacetyl coenzyme A is catalyzed by acetoacetyl coenzyme A thiolase. Okay, and so this reaction is reversible. And so note that this reaction is reversible. Okay, your acetoacetyl-CoA, and so will condense with another coenzyme A, and so to produce you with your acetyl co and so your acetoacetyl coenzyme A will condense with another acetyl coenzyme A to produce a beta hydroxy beta methyl glutaryl coenzyme A. Okay, and so you have there. And so a condensation of your acetoacetyl coenzyme A with another acetyl coenzyme A will produce beta hydroxy beta methyl glutaryl coenzyme A. Okay, and so in this reaction is catalyzed by your beta hydroxy beta methyl glutaryl coenzyme A synthase. Okay, and so this reaction is catalyzed by your beta hydroxy beta methyl glutaryl coenzyme A synthase. Okay, and this beta hydroxy beta methyl glutaryl coenzyme A, and so is cleaved and so to form your acetoacetate and acetyl coenzyme A. Okay, and so your beta hydroxy beta methyl glutaryl coenzyme A, and so is cleaved, and so to form acetoacetate and your acetyl coenzyme A. And so you already have your acetoacetate here as a ketone body. Okay, and so and this one, and so this the production of your acetoacetate and your acetyl coenzyme A is catalyzed by your beta hydroxy beta methyl glutaryl coenzyme A lyase. Okay, and so with that. So your acetoacetate and so is reduced to form your beta hydroxy butyrate. And so your acetoacetate is reduced to form your beta hydroxy butyrate. And so note that there is a reduction that happens. Okay, and so that has take place. And so in the production of your beta hydroxy butyrate. And so with this, it is reversible. And so this reaction is reversible. And so what you call this, there if there is an increased concentration of your acetoacetate, and so it will produce acetone, and so via the carboxylation, and so release of your carbon dioxide here. Okay, and so there is a release of your carbon dioxide, and so from your acetoacetate to produce your acetone, and so via your what you call this, spontaneous decarboxylation, and so to produce your acetone again. And so when there is an increased concentration of your acetoacetate, and so it will produce, and so it will undergo spontaneous decarboxylation, and so a release of your carbon dioxide to produce your acetone, of course. And so with this accumulation of ketone bodies in the blood and tissues, and so and so uh will cause discomfort to you. Ayan, so with that, and so in this one is seen, and so accumulation of your ketone bodies in the blood and tissues, ayan, is seen in cases of prolonged starvation and uncontrolled diabetes. And note that when there is an increased accumulation of your ketone bodies in the blood and tissues, ayan, so this can lead to serious effects. Okay, and so if not treated right away. Okay, and so let's take a look at your ketone bodies. And so you have here your acetoacetic acid or your acetoacetate, and so it is 20% of your total, no? And then your beta hydroxybutyric acid or your beta hydroxybutyrate, and so 70% of your total, and then your acetone at 2%. Okay, and so that is for your ketone bodies. Okay, and so your several tissues accordingly, and so they can utilize your ketone bodies for energy production. And so normally, ayan, so they can use ketone bodies to generate energy. Okay, and so you have there. And so of course, and so during prolonged starvation, and so in the that is in the absence of sufficient glucose, and so your brain and so uses ketone, and so your brain uses ketone bodies as energy source, no? And so in case of prolonged starvation. Okay, and so you have there. And so you have, of course, and so with that, and so others, you have your heart and your skeletal muscles, or your cardiac and your skeletal muscles, and so utilize your they can utilize uh ketone bodies, and so as an energy source under normal conditions. Okay, and so you have there. Okay, and so also, and so one important thing here is, and so your oxidation of your ketone body, and so will spare your skeletal muscle proteins, and so it will what you call this, save your skeletal muscle proteins, and so instead that it will be broken down, it will undergo gluconeogenesis, and so to produce glucose 6 phosphate, and so it is spared, and so to produce by your ketone body oxidation. Okay, and so let's take a look at also, ayan, so your enterocytes, sorry, and so your enterocytes and your adipocytes, and so they can also use your ketone bodies, and so to produce energy. Okay, and so you have here, and so let's take a look at your utilization of this one. Okay, and so your beta hydroxybutyrate and so your beta hydroxybutyrate, and so is converted into your acetoacetate. And so this one actually is your beta hydroxybutyrate here. And so this one is your ketone body. And so it will be utilized, and so to produce energy. And so your beta hydroxybutyrate, and so is converted to your acetoacetate, and so that is catalyzed by your enzyme beta hydroxybutyrate dehydrogenase. And so in this reaction, and so there is a formation of your NADH, and so your NADH here, and so there is the formation of your NADH. And so note that in the utilization of your ketone bodies for your energy, and so you have your beta hydroxybutyrate and your acetoacetate only. And so your acetone accordingly is not utilized for energy production. Okay, and so still, and so under your utilization of your ketone bodies for energy, and so you have your acetoacetate, and so is converted into your acetoacetyl coenzyme A. And so this what you call this reaction, and so your conversion of your acetoacetate to your acetoacetyl coenzyme A is catalyzed by beta ketoacyl coenzyme A transferase, no, and so that is your beta ketoacyl coenzyme A transferase, and this one is absent in the liver. Ayan, so your beta ketoacyl coenzyme A transferase is absent in the liver, and so that is why, and so your liver use cannot utilize your ketone bodies as energy. Again, and so they say beta ketoacyl coenzyme A transferase is absent in the liver. That is why your liver cannot utilize ketone bodies as a substrate. I mean, as a source for energy. Okay, and so note that your acetoacetate and your beta hydroxybutyrate, and so they are the uh, ketone bodies that are utilized in the production as a source of energy. Okay, and so in this case, and so you have to use your succinyl coenzyme A to activate your acetoacetate, and so into acetoacetyl coenzyme A, and so you need your succinyl coenzyme A, and so to activate your acetoacetate to produce acetoacetyl coenzyme A. And so you have there this succinyl coenzyme A is converted naman into your succinyl only. Okay, and so that is for that. And then, and so your acetoacetyl CoA, and so is converted into your acetyl coenzyme A, that is catalyzed by acetoacetyl coenzyme A thiolase. Okay, and so your acetoacetyl coenzyme A is converted to your acetyl coenzyme A, that is catalyzed by acetoacetyl coenzyme A thiolase. Okay, and so let's take a look at the energy yield from the catabolism of your beta hydroxybutyrate. Okay, and so let's take a look here. And so you have here your you have produced two acetyl coenzyme A accordingly. And so you produced two acetyl coenzyme A in the process. And so with this, and then you also produced one NADH. Okay, and so you produce one NADH. And so by the oxidation of your beta hydroxybutyrate to form your acetoacetate. Okay, and so you have one in ADH that is produced. Okay, and so and then you used actually, and so you deduct, and so from that you deduct 1 ATP, and so that is used from the activation of your acetoacetate by your succinyl coenzyme A. And so you used 1 ATP there, and so with that, of course, you calculate accordingly again, and so 2 acetyl coenzyme A, and so will produce 10 ATP accordingly, and so it will produce 280 10 ATP, one acetyl coenzyme A will produce 10 ATP. And so in that is in the citric acid cycle and the electron transport chain and your oxidative phosphorylation with that. Okay. And so with that still you have energy yield. And so you can still use accordingly your ketone bodies as a source of energy. So with that, let's take a look at your ketogenic and your anti-ketogenic. Okay, and so your ketogenic, accordingly, these are substances which form ketone bodies. And so you have your fatty acids and your isoleucine, leucine, your phenylalanine, and your tyrosine. While your anti-ketogenic, and so you have there your carbohydrates and of course your glucogenic amino acids. Okay, and so let's take a look at your oxidation of your fatty acid, unsaturated fatty acids, no? And so the oxidation of these unsaturated fatty acids requires additional enzymes. Okay, and so note that most of your fatty acid, your unsaturated fatty acid, they have a cis. configuration. And so they have a cis configuration. And so unlike the trans double bonds that are introduced during the beta oxidation of your saturated fatty acids. Okay, and so again, note that your naturally occurring unsaturated fatty acids, they have a cis configuration. Okay, and so you need to what you call this, to you need to convert the cis into a trans. Okay, and so with that, you need your enzyme, enol coenzyme A isomerase, and to convert your cis beta gamma double bonds to trans alpha beta double bond. Okay, and so you have here, you have your cis beta gamma double bond, and so to trans alpha beta double bond. Okay, and so note that you have there as your what you call this as the site of the reactions for that. And so your alpha and beta. And so you need your enzyme enol coenzyme A isomerase. And so to convert your cis beta gamma double bond to your trans alpha gamma double bond. Okay, and so example of your unsaturated fatty acids that require additional enzyme. And so your unsaturated fatty acids, of course, all of them require additional enzymes. And so examples of which of unsaturated fatty acid, you have your oleic acid and the rest. Okay. And so you have, if you have an even numbered fatty acids, you have actually odd. And so, but then most of what you call this, your for humans, and so you have your even numbered. You have at 18, at 20, those items, 20 carbon atoms. But then you have your odd chain fatty acids. Let's take a look at this for real quick. Okay, and so your odd chain, and so they contain what you call this, your odd numbered, no? And so they accordingly occur in some plants, no, and microorganisms. Okay, and so you have here beta oxidation, and so beta oxidation also takes place in this one, but then, and so what you call this, it will yield, no? And so it will yield one acetyl coenzyme A and one propionyl coenzyme A molecule. Okay, and so and this one, of course, and so the beta oxidation proceeds normally to produce your one acetyl coenzyme A and one propionyl coenzyme A, and then this propionyl coenzyme A is converted to your succinyl coenzyme A. And so this one is converted into your succinyl coenzyme A. And note that your succinyl coenzyme A is a citric acid cycle intermediate. Okay, and so again, and so this one is for what you call this odd chain fatty acid oxidation, and so takes place in some plants and animals. And so but then and so accordingly, and so not in uh and so it takes place in some plants and animals. Okay, and so let's take a look at your fatty acid synthesis. And so this one occurs mainly in the liver. And so synthesized, and so this your fatty acids are synthesized when the diet is low in fat, or there is a there is high in, or the diet is high in carbohydrate or protein, and that is the diet is low in fat, no? And so you have there, and so accordingly, and so your fatty acid synthesis and so occurs within the cytoplasm of your most of the animal cells. But then the major site is uh, the liver still, and so the liver is the major site for your fatty acid synthesis. Okay, and so most of your fatty acids are synthesized from excess dietary carbohydrate, as what we said earlier. And so as the first intermediate, and so you have your malonyl coenzyme A. No, and so it requires a large amount, and so your fatty acid synthesis requires large amount of your NADPH, and so requires large amount of your NADPH. And so that is of course you for your supply of this NADPH is from your pentose phosphate pathway. And so pentose phosphate pathway. And so the reactions accordingly that are catalyzed by your isocitrate dehydrogenase and your malic enzyme, and so provide smaller amount. And so but then, and so the major source of your NADPH for this is your pentose phosphate pathway. Okay, and so in your fatty acid synthesis, accordingly, this one appears to be a reverse of your beta oxidation. Again, as what we said earlier, and so it is predominantly in the cytoplasm. And so this fatty acid synthesis occurs predominantly in the cytoplasm. And so let's take a look at this fatty acid synthesis. Okay, and so you have there. And so actually, this fatty acid synthesis occurs in two phases. And so the first phase is your carboxylation of your acetyl coenzyme A to form your malonyl coenzyme A, and so that is catalyzed by your acetyl coenzyme A carboxylase. And then, of course, the synthesis of your palmitate by the sequential addition of your carbon units. No, and so that is the second one. Okay, and so for the first one, the carboxylation of your acetyl coenzyme A to form your malonyl coenzyme A by your acetyl coenzyme A carboxylase. And the second, and so second phase is the synthesis of your palmitate, synthesis already of your fatty acid by your sequential addition of your two carbon units, and so to the growing fatty acid chain by your fatty acid synthase. And so in the second phase, of course, you have there your fatty acid synthase as the key enzyme. Okay, and so your fatty acid synthesis. Okay, and so with this, and so and so it starts, and so with your what you call this, when your acyl carrier protein, and so or this one is your ACP, your acyl carrier protein combines with acetyl coenzyme A and malonyl coenzyme A to form your acetyl uh, acyl carrier protein and your malonyl acyl carrier protein accordingly. Okay, and so this one is catalyzed by your malonyl acetyl transferase. Okay, and so this reaction, your acyl carrier protein and the combination of the condensation of your acyl carrier protein and your acetyl coenzyme A and your malonyl coenzyme A is catalyzed by your malonyl acetyl transferase. Okay, and so your malonyl acid malonyl is ACP and your acetyl ACP, and so will condense to form your acetyl ACP. And so they will combine, no? And so in this one, this reaction is catalyzed by your ketoacyl synthase. Okay, and so your acetoacetyl ACP, and so is converted to your beta 3-hydroxybutyryl ACP. Ayan, so this is catalyzed by your beta ketoacyl ACP reductase. Okay, and so your beta 3 hydroxybutyryl ACP, and so is converted into your crotonyl ACP, and so that is catalyzed by beta hydroxyacyl ACP dehydratase. Okay, and so with that, and so in this reaction, and so there is a formation of your carbon to carbon double bond by removal of water. Okay, and so carbon to carbon double bond by removal of water. And so your crotonyl ACP, and so is converted to your butyryl ACP. And so your butyryl ACP, and so this one is catalyzed by your enol ACP reductase. Okay, and so that is catalyzed by your enol ACP reductase. And so of course, and so in your your enol ACP reductase, and so will produce your saturated 4 carbon acyl group that is actually in this reaction, and so in your reaction of your crotonyl ACP to butyryl ACP, and so your butyryl ACP here is a saturated 4 carbon atom acyl group. Okay, and so you have there in this acyl group, and so this acyl group is transferred from your ACP to your SH group of your ketoacyl synthase. Okay, and so your ketoacyl synthase. And so this one, and so the acyl group is transferred from your ACP to the SH group of your ketoacyl synthase, and so to begin a new elongation cycle. And so this one, and so to begin a new elongation cycle. And so this one actually is a 6 elongation cycle. Okay, and then of course your acyl chain will lengthen, and so by 2 carbon atoms as it will condense with another ACP link malonyl group. Okay, and so with that, and so your fatty acid synthesis will uh, end, and so with the release of your palmitate from your ACP, and so that is catalyzed by your thioesterase. Okay, and so that is catalyzed by thioesterase. Okay, and so let's take a look at your fatty acid synthase. And so your fatty acid synthase, and so you have here this one is what you call this one, and so your fatty acid synthase, and so it will catalyze the remaining reactions in the fatty acid synthesis. Okay, and so this one actually is your fatty acid synthase is in an X-shaped. And so X-shaped, and so head-to-head homodimer composed of two identical polypeptides. Okay, and so it has two identical polypeptides, and so with that, and so each polypeptide has two catalytic domains and ACP, and so it has two catalytic domains and ACP, and so with that, and so it has two catalytic domains, and so and two ACP, and so your fatty acid synthase, and so can synthesize two fatty acids simultaneously. And so it will, what you call this, it can synthesize two fatty acids simultaneously. And so, and then of course, and so, once you have here, once your, what you call this, once you already have your, enough, the fatty acid has already lengthened, and so, and so uh, reach its length, and then you have here your fatty acid is cleaved by your thioesterase, and so it is cleaved by your thioesterase. And so let's take a look at the metabolism of your phospholipids. And so your phospholipids, and so your components, and so one phosphoric acid, one glycerol, and two fatty acids with one nitrogenous base. Okay, and so that is in your plasma membrane, and so located in your plasma membrane. And so you have there, and so with this, and so important phospholipids, you have there your lecithin and your sphingomyelin accordingly. And so you already know this. Okay, and so your absorption of your phospholipids, and so your dietary and your endogenous phospholipids may be absorbed as such, as phospholipids, or can be hydrolyzed by your lecithinase from your pancreatic juice. Okay, and so let's take a look at your metabolism of this. And so your lecithin, and so is converted into your fatty acid to your glycerol, phosphoryl choline. And then your cephalin, and so that is converted to your fatty acids plus your glycerol, phosphoryl cholamine, or your ethanolamine with that, no? And so your glycerol phosphate here, and so maybe your glycerol phosphate linkage here may be hydrolyzed in the intestinal mucosa by your phosphodiesterase, and to break down the phosphate linkage. Okay, and so let's take a look at your phosphoglyceride synthesis in the endoplasmic reticulum. And so you have your de novo synthesis, in which you have your phosphatidylserine serves as a precursor for other phosphatides. No, and so your, what they call this, your CDP, diacylglycerol, and so that is your common precursor of your phospholipids. Okay, and so your phosphatidylserine, and so that is equal to your phosphatidylethanolamine and to your phosphatidylcholine. And so they have actually same synthesis for this phosphatidylserine, and so they have same. And then of course your utilization, and so of your exogenous choline, and so in your salvate, this one your choline, and so this one is the base component of your phospholipids, and of course, and so needed for the synthesis of your lecithin. Okay, and so let's take a look at your sphingolipids. Okay, and so your sphingolipids metabolism, and so this your sphingolipids, and so they contain your ceramide. No, and so they contain your ceramide, and so contains your ceramide derivative of your amino acid. This ceramide, and so is a derivative of your uh, of your amino acid alcohol sphingosine. Okay, and so let's take a look at your ceramide synthesis. And so your ceramide, and so your synthesis begins with the condensation, the combination of your palmitol coenzyme A with your serine, and so to produce your ketosphingolene, no? And so, of course, that reaction, and so is catalyzed by 3-ketosphingolene, and so 3-ketosphingolene synthase. And so this 3-ketosphingolene, and so is reduced by your NADPH to sphingolene. And so your 3-ketosphingolene and so is reduced by your NADPH to produce sphingolene. Okay, and so your sphingolene and so is converted to ceramide into two process. And so of course it will involve your acyl coenzyme A and your FADH2. And so the two process will involve your what you call this acyl coenzyme A and your FADH2. Okay, and so you have there. And so your ceramide, and so when your ceramide reacts with phosphatidylcholine, and so it will produce sphingomyelin. And so when your ceramide reacts with your UDP, uridine diphosphate glucose, and so to produce your glucosyl transferase. And so when it reacts with your UDP, it will produce your glucosyl cerebroside, sorry. And so it will produce your glucosyl cerebroside or your glucosyl ceramide. Okay, and so you have there. Okay. And so in your production of your sulfatides, and so your sulfatides actually are synthesized when your galactocerebroside, and so reacts with a sulfate, no? And so with that, okay. So let's take a look at your cholesterol synthesis. Okay, and so your cholesterol, and so can be derived in two sources, and so endogenous and exogenous. Of course, that is this one, your cholesterol can be synthesized by the liver and then, of course, from the diet. And so, with that, and so your synthesis is what we call this depressed, and so when there is enough in the diet, and so usually, and so your daily requirement is at least 400 milligrams per day. And so if you reach such, and so your body can will not synthesize accordingly cholesterol. And so you have there, and so but then, and so you have here, and so you have average 900 milligrams per day that is actually produced. Ayan, ayan. So your cholesterol, ayan, so average, your cholesterol synthesis, ayan, so is about 900 milligrams per day. And so, and also, ayan, so your LDL receptor synthesis, these are stimulated when, what you call this, the diet is low in cholesterol. Okay, and so a biosynthesis of your, what you call this, cholesterol and your LDL receptor, and so is stimulated when the diet is low in cholesterol. Okay, and so with that, okay, and so with that, there is a formation of your beta hydroxy beta methyl glutaryl coenzyme A, and so from your acetyl coenzyme A. And so this one in the synthesis already of your cholesterol. And so your acetyl coenzyme A, this one is a precursor for your synthesis of cholesterol. Okay, and so your acetyl coenzyme A, and so is convert, I mean, will form your beta hydroxy beta methyl glutaryl coenzyme A. And this one, ayan, so you have there. And then, ayan, so, your the next step, and so in the cholesterol synthesis is your is what you call this, the conversion of your beta hydroxy beta methyl glutaryl coenzyme A to your squalene. And so your squalene, and so the third step, and so is your squalene is converted into your cholesterol. Okay, and so you have there. And then, and so let's take a look at your down regulation of this, your cholesterol. And so, unlike, and so your biomolecules like your uh, light triacylglycerol. And so your yes, and so they can be what you call this degraded and so into smaller molecules. But then your cholesterol and so accordingly cannot be degraded into smaller molecules. And so that is why, and so there is no degradation that will take place for your cholesterol. You only have to do your down regulation of the synthesis, and so to maintain normal, to maintain balance. And so you have your down regulation of the synthesis of your cholesterol. And so this is your bile acid synthesis. This one is a disposal pathway for your cholesterol. And then, of course, bile acid synthesis and excretion. And then, of course, your synthesis of your steroid hormones. And so this one, and so they say down regulation of the synthesis, and so bile acid synthesis and excretion, and then your synthesis of your steroid hormone, and so they combine to lower circulating cholesterol levels. And so this one again, and so down regulation of the synthesis of your cholesterol, the bile acid synthesis and excretion, and then yours what do you call this, and then your synthesis of your steroid hormone, all of this will contribute to lower your circulating cholesterol levels. Okay, and so note that in the synthesis of your bile acids, you have it takes about one half, and so one half of your cholesterol that is synthesized daily, and so is used to synthesize bile acids. Okay, and so they are used to synthesize bile acids. And so bile acids, they are synthesized and so in the liver. Okay, and so you have there. And so conversion of your, and then of course you have your conversion of your cholesterol to 7. This one actually is for your synthesis of your bile acids, no? And so you have your conversion of your cholesterol to your 7 alpha hydroxy cholesterol. Okay, and so the conversion of your cholesterol to your 7 alpha hydroxycholesterol, and so this one is catalyzed by your cholesterol hydrolase, and so is the rate limiting reaction in the bile acid synthesis. Okay, and so your conversion of your cholesterol to 7 alpha hydroxycholesterol cholesterol, and so is the rate limiting reaction in the bile acid synthesis, and so with that it will actually stop, no, and so the synthesis will stop. And so that your this reaction is catalyzed by your cholesterol 7 hydroxylase. Okay, and so note of the importance of your bile acids, and so they are important in emulsification and probably, and so that's fat digestion. Okay, and so with that, and so of course in your in your uh balance of your cholesterol, and so you have there your what you call this uh your this one is your balance is achieved through the mechanisms that regulate your biosynthetic pathway. And so you have your also, and so it will also regulation also of your LDL receptor activity. Ayan. And then of course the bile acid biosynthesis, and so this all of this will contribute to the balance of your cholesterol, and so the homeostasis of your cholesterol. And so one important thing, and so in the regulation of your cholesterol biosynthesis, and so is the modulation of your existing HMGR molecules. And so this one is your HMGR. And so you have there, and so the regulation again of your cholesterol biosynthesis, and so can be accomplished by modulation of your existing what they call this 3-hydroxy 3-methylglutaryl coenzyme A molecules, and so coenzyme A reductase molecules. Okay, and so with that, okay, and so and so it is important, and so to regulate, and so to keep your cholesterol levels at a normal, I mean at a normal levels. Okay.